Alzheimer's disease is a devastating brain disorder that affects millions of people around the world. It slowly robs individuals of their memories, cognitive abilities, and ultimately, their independence, making everyday tasks increasingly difficult. As the most prevalent form of dementia, Alzheimer's affects millions worldwide, with numbers expected to rise as the global population ages. This progressive neurodegenerative disease poses a significant challenge to healthcare systems and families alike, requiring extensive care and support. Understanding the mechanisms behind this complex disease is crucial for developing effective treatments and improving the lives of those affected. Researchers are tirelessly working to uncover the mysteries of Alzheimer's. The human brain is a complex organ responsible for our thoughts, memories, and actions. It is composed of billions of neurons that communicate through intricate networks. In Alzheimer's disease, this intricate network is gradually destroyed, leading to a decline in cognitive function and the loss of essential brain activities. The disease progresses through distinct stages, each marked by worsening symptoms. Early detection and intervention can help manage the progression. Early signs may include subtle memory lapses and difficulty finding words, often mistaken for normal aging. As the disease advances, individuals experience more profound memory loss, disorientation, and changes in mood and behavior, which can be distressing for both the patient and their loved ones. The exact causes of Alzheimer's disease are still not fully understood, but ongoing research is shedding light on potential factors. However, scientists have identified several key players and processes that contribute to the development and progression of the disease, including genetic, environmental, and lifestyle factors. These include the abnormal accumulation of certain proteins in the brain, leading to the formation of plaques and tangles, which are hallmarks of Alzheimer's. These toxic aggregates disrupt communication between brain cells and ultimately lead to their death, causing the brain to shrink over time. In the following sections, we will delve deeper into the intricate mechanisms underlying Alzheimer's disease, exploring the latest scientific discoveries. We will explore the formation of amyloid plaques and tau tangles, which are critical in the disease's progression, the role of chronic inflammation, which exacerbates the damage, and the devastating consequences of synaptic loss and neuronal death, which lead to the cognitive decline seen in patients. Furthermore, we will examine the genetic and environmental risk factors that contribute to the development of this devastating disease, including lifestyle choices and family history. Lastly, we will discuss current therapeutic approaches and promising research avenues that offer hope for the future, including new drugs, lifestyle interventions, and potential cures on the horizon. One of the hallmarks of Alzheimer's disease is the accumulation of amyloid beta plaques in the brain. These plaques are abnormal clusters of protein fragments that disrupt the normal functioning of brain cells. These plaques are formed from a protein called amyloid precursor protein, or AP, which is normally found in the membranes of brain cells. AP plays a crucial role in the growth and repair of neurons. APP is broken down into smaller fragments, one of which is amyloid beta. This process is a normal part of cellular metabolism. In healthy brains, amyloid beta is cleared away efficiently through a series of complex mechanisms involving enzymes and immune cells. However, in Alzheimer's disease, this clearance process is impaired, leading to the buildup of amyloid beta in the spaces between neurons. This accumulation is a gradual process that can take years or even decades. These amyloid beta fragments stick together, forming clumps called oligomers. Oligomers are small, soluble aggregates that are particularly toxic to neurons. These oligomers then aggregate into larger structures known as plaques. Over time, these plaques grow and spread throughout the brain. The presence of amyloid plaques disrupts communication between neurons. This disruption is due to the plaques physically blocking the pathways that neurons use to send signals to each other. These plaques interfere with the transmission of signals across synapses, the junctions between nerve cells. Synapses are critical for memory and learning, and their impairment leads to significant cognitive decline. This disruption impairs cognitive function, leading to the memory loss and other cognitive impairments characteristic of Alzheimer's disease. Patients often experience confusion, difficulty in problem solving, and changes in behavior. The amyloid hypothesis, which posits that the accumulation of amyloid beta is the primary cause of Alzheimer's disease, has dominated the field for decades. This hypothesis has driven much of the research and therapeutic strategies aimed at treating Alzheimer's. However, while amyloid plaques are a consistent finding in the brains of individuals with Alzheimer's, they do not fully explain the complexity of the disease. Other factors such as tau protein tangles, inflammation, and genetic predispositions also play significant roles in the progression of Alzheimer's. Another key player in the pathogenesis of Alzheimer's disease is the tau protein. This protein, which is found in neurons, plays a crucial role in maintaining the stability and function of the brain's communication network. Tau is a protein that helps stabilize microtubules, which are essential for transporting nutrients and other important molecules within neurons. Microtubules act like highways inside the cells, ensuring that everything reaches its proper destination. In Alzheimer's disease, tau protein becomes abnormally modified. This abnormality is one of the hallmarks of the disease and plays a significant role in its progression. It undergoes a process called hyperphosphorylation, where it accumulates an excessive number of phosphate groups. This excessive phosphorylation alters the normal function of tau. This hyperphosphorylation causes tau proteins to detach from microtubules and clump together, forming neurofibrillary tangles inside neurons. These tangles are toxic and interfere with the cell's internal transport system. These tangles disrupt the transport system within neurons, hindering their ability to function correctly. The disruption means that essential nutrients and molecules cannot be transported efficiently. The disruption of microtubule function by tau tangles can have devastating consequences for neurons. The cells become damaged and are unable to communicate effectively with each other. Without a functional transport system, neurons are unable to receive the nutrients and essential molecules they need to survive. This deprivation leads to a cascade of negative effects within the brain. This can lead to neuronal dysfunction and eventually cell death. The loss of neurons is a critical factor in the cognitive decline seen in Alzheimer's patients. As neurons die, the brain shrinks and cognitive function deteriorates further. This shrinkage is often visible in brain scans of individuals with advanced Alzheimer's disease. The interplay between amyloid plaques and tau tangles is complex and not fully understood. Researchers are still working to unravel the exact mechanisms behind this interaction. However, it is clear that both contribute significantly to the development and progression of Alzheimer's disease. The presence of both these pathological features is a defining characteristic of the condition. The presence of amyloid plaques is thought to trigger the hyperphosphorylation of tau, leading to the formation of tangles. This suggests a possible sequence of events where amyloid pathology precedes tau pathology. This cascade of events ultimately results in neuronal dysfunction and death. Understanding this sequence is crucial for developing effective treatments and interventions for Alzheimer's disease.